Hi, everybody. Can everyone hear? I think it's time for us to begin so we don't run out of time with our lecturer and our readings this evening. I'm Linda Jillison. Again, as usual, I get to introduce our speaker and our readers and welcome all of you to this wonderful evening and our 17th, I believe, annual Alumni Association lecture series. First of all, you know what I always ask, stand up here and I'm going to ask you to turn off your telephones if you haven't already done that. And then just remind you about a couple of things. As usual, we'll be having a little celebration after the last lecture just down the hallway, so prepare for a wine and cheese or something like that. And secondly, I want to let you know that I just have the schedule for the MCAT broadcast of all of these lectures. And the first will be on March the 30th, and each one of them seems to be broadcast twice. And then they will end with the last one, Emily Grassley's, on May 4th and May 8th. And Susan's also asked me to tell you that there will be series DVDs available to order. We're hoping that we'll be able to give people a chance to order them at least at the last evening of the series, and then we'll be able to let you know when your DVD is available. So if you're really having a great time with this series, you'll be able to take a DVD home and watch it in your downtime at home, because I think it's been wonderful thus far, and I look for it to continue in that same way. As usual, I'm going to introduce pretty quickly here Jeff Badenoch, who will be doing the translation of the poem, which was written in Spanish by my colleague Eduardo Chirinos, and Eduardo will read first. First of all, I want to mention to you that again with us, David Mile Lusk is here. David, would you stand up, please? David's the artist for Eduardo's book. We're really pleased to have you here. Thank you for coming. First of all, we have Eduardo Chirinos. Eduardo is, as I said last week, one of my colleagues in the Department of Modern and Classical Languages. He's a world-famous poet, and we've had students go and study abroad and come back from who knows where and say they found a copy of Eduardo's books on a bookshelf in a railroad station or something. People in different countries read different kinds of railroad books from us, I'll tell you, you know. But in any case, we're really, really so pleased to have Eduardo as a colleague. He's a wonderful creative artist and a great person to be with. And I want to introduce also, Janine, would you stand up, please? This is Janine Montauban, who's one of our chair people in the Department of Modern and Classical Languages and Literature, and she's married to Eduardo, and the two of them are a great pair. So first I'm going to have Eduardo come over and read his poem, and then Jeff will follow up, and then I'll get a chance to introduce Creagh. So Eduardo and Jeff.
Good evening. Tonight I will read a poem whose speaker is not me. It's this bird, Chersophilus duponti, maybe the most famous bird in the literature, the lark, or in British English, the skylark. Maybe most of you remember the Shelley's poem, To Skylark. Chersophilus duponti. ¿Por qué negarlo? Soy un ave favorecida por la literatura. Según los manuales, mi área de dispersión es Europa y gran parte de África. Como no soy migratoria, de allí no me muevo, tampoco de las páginas que me han dedicado los poetas. ¿Quién no recuerda aquello de «Cambei la lauceta, mover de yo esas alas contra el ray»? Eso lo escribió un trovador que envidiaba mi alegría, la dulzura de mi corazón cuando volaba. Podría citar más ejemplos y todos dirían lo mismo, que mi canto es un don del universo, que esconde los secretos del amor, que expresa las dentillas del deseo. Todo muy halagador y muy bonito, pero no me sirve de mucho. Ahora, mi canto solo se escucha en el norte de África, en los secanos áridos del Valle del Ebro, donde soy conocida con el nombre de Rosín. Muchas gracias.
Why deny it? I've been a bird literature has favored. According to textbooks, I can be found throughout Europe and much of Africa. I'm not migratory, so I don't move much from there or from the pages poets have dedicated to me. Who doesn't remember the lines, Canvai la lozueta mover de hoy sas alas contradrai? A troubadour who envied my happiness wrote that, jealous of the sweetness of my heart in flight. I could cite more examples, but they'd all say the same things, that my song is a gift from the universe, that in it dwells the secrets of love, that it expresses the sorrow of desire. This is all very flattering and nice, but doesn't do me much good. Today, my song can be heard only in northern Africa and on the arid sandbanks of the Ebro Valley, where I'm known in Spanish as the hack and the nag.
Thanks, both of you, very, very much. Eduardo and Janine have to leave. They have another obligation, so. Now I got a chance to introduce Creagh Breuner. Creagh, where are you? Oh, there she is in the very back. All right, so I'm glad you found a seat. Creagh and I actually got a chance to, at Creagh's suggestion, sit down and have a chat this past week for a little bit, and it was great fun. She came over to my office, and we just chatted about what she does here at the university and how long she's been here and what kind of things she likes to do with her time. We actually found out that we both have a life outside the university. Hers is musical, mine is political, but we share some common friends, so that was a lot of fun. It's good to know there's life out there. Creagh actually came to us in 2006, and I should correct what's in the program and say that she's now a full professor. She was just recently promoted, so congratulations, always good. And she teaches in the wildlife biology program. She works with students of various kinds, students who are going to be in medical school, students who are going to be in veterinary school. And her interest really is in stress and stress hormones and how they affect the behavior of birds. Something which probably a lot of us will want to know about and whether her findings transfer to human beings or not. So listen up. She seems to have a wonderful combination of working in the lab and working outdoors. She's done research not only in the lab, where she has graduate students with whom she works, but also in Hawaii and in the San Juan Islands, and also up by Ronan, I believe. And so she's one of those people who really lives out this title of our lecture for tonight, of our series for this year, in fact, and that is Birds Inside and Out. So I'm going to ask Creagh to come on over and chat to us about stress. It's not all bad. Creagh?
All righty, thank you. All right, so this is on, and if I go this, you can hear me. When I'm talking like this, can you still hear me? Am I good? Thanks, Eric. Okay, so I'm really excited to be here tonight. I'm excited to talk to you guys about things that I care a lot about. And I'm excited to maybe change the way you view what's happening in the world, if even just a little bit. But to start with, we're actually going to do something. Let me get all my equipment together, do that. We're going to try something a little bit different first. So I'm going to take out of this. And that pops up, which I'll use in a second. So if I come over to this group, you guys over here, how many of you guys are comfortable taking your pulse? If I were to give you about 10 seconds, okay. So you guys in the back can do it too. I'm going to just, well, I'll give you 10 seconds, take your pulse. So go ahead. About halfway there. Okay, So why don't we get, and I need to be able to see while I'm typing what I'm doing. So I'm going to do this for a second. And get my mouse onto here. That will be interesting. Oh, there we are. All righty, so can I get a couple, what are a couple estimates over here? Don't multiply it by 6. We don't have to do any math right now. What's you? 11? 12, 12, 10? Awesome. Okay, I got to get some of those in. So we got 11, 12, 12. We got a 7. We got a 10. Let's get 3 or 4 more. 15, 11, 12. Man, 10. Okay, so 15, 11. Oh, I'll change that one. Because that's really not going to help the data. And already, I think I heard one more 15. So let's set that, and we can actually go over here, and it's a nice thing we can see where all you guys fit on there with the mean for those people. And then this is just a demonstration, so no pressure, but I'm going to ask you to do a little math.
What is this math of which you speak?
Yes, so what's 3 times 6?
18,
So we get a little bit harder. What's 5 times 12?
60,
Okay, then how about 6 times 2.5?
13.
I get 15.
Oh,
with
wait a
that
minute, you're
one,
right.
but that's
There you
okay.
go. Yeah, I see it.
So we'll go there. So let's see, how about you, sir? You want to do some math for me? What's the square root of 9?
3,
Okay, square root of 16.
4,
So this test that we're doing here is actually something called the Creagh social stress test. And what they actually ask people to do is to start at 1,022 and count backwards by 13s. You want to try one of them? Excuse me for crowding you.
1009,
22. Yeah. Next one's hard.
997,
I get 996. Here, look at this. We'll get one more. Do you feel like helping me out on this one?
Jeff can, Bill can.
Jeff, there.
I've already worked.
I have already worked. Actually, well, did you give me a pulse rate at the beginning?
No, I didn't.
Okay. Then, okay. So actually, we're going to do this again. We're going to take your pulse rate. So let me do that. And you guys ready? Okay. Go ahead. All right. Stop. All right. In this group here, what did we get? 11, 15. I was wondering if this was going to work or not. 13, 14, 11. So we go back up here, and we go 11. Oops, that's wrong. 13, 14. Oh, the 8. 12. Alrighty, yeah, exactly. That's someone who does. So I wasn't sure how this would work. It's up a little bit. What I did in case this didn't work, because I wasn't really going to, when they do this, this is actually, so this is the Creagh social stress test. And it's a way to apply a standardized stressor that has set rules that they have people go through. And psychologists use this to evaluate how someone responds hormonally and psychologically to stress. And actually, what you do is you have to start at 1,022, count backwards from 13, and the panel of people watching you don't smile and don't give you any, oh, that's okay. And they actually, it's this very standardized, very serious thing, and I'm not that kind of person, so I couldn't quite do that. But what I did do was look up a study where this was published in 2008, where there was some heart rate data, and this is again per 10 seconds. And what you can see is there's an increase. So heart rate goes up even though you're just sitting there. You're not getting up and running around the block. You're not doing anything that would require more oxygen to your cells, more glucose to your cells. So what is that? Well, that's what I'm going to talk about today, which is stress. And let's get me back in order and get my things back in order. And I can go back to the talk part. But I have to get back onto this one now, and I need my glasses for that. So I am still up there. Am I still? I am. There I am. There I am. What'd you say? Oh, my heart rate's been high all week. We're good. All righty. So what is stress? So stress is a pretty nebulous word. It means a number of different things. And I want to just start out defining what stress is. Stress primarily means two different things. One is the stressor. It's the event that causes stress. But it's also thought of as the condition. What's happening inside the animal that the stress is happening to. So let me walk you through some kind of definitions for this to help us move along. When we think about stressors, there are stressors for wild animals living in their natural environments, and there are stressors for humans. We can have, bad weather, cold temperatures, social interactions, predation, there's a stressor. We can have parasites, injury, low food availability. In humans, social interactions can be stressful. Worrying about money is a huge stressor in humans. Having to study for exams, worrying about exams. And it ends up that all of these examples of stressors tend to fit into three separate categories. So there are physical stressors, physiological stressors, and emotional stressors. Physical stressors is what you think. It's an injury. It's a physical change to your body. Physiological stressors, when there's bad weather and animals get too cold, they get too hot. When there's low food availability and there's no nutrients, that's a physiological stressor. But then we get into the emotional stressors, and those are the zingers in humans. So animals have psychological stressors, so dominant-subordinate interactions. That's a big one. That's one of the main psychological stressors you'll find in wild animals. An increase in predators, so increase in predation pressure, that's a psychological stressor. But really what we could identify in this room is not being able to get enough food for our families, having to worry about money, having to study for exams. Those things are emotional stressors, and that's what the math was here, if I had been a little meaner. That we potentially, that's not, you guys aren't getting up and reducing your glucose in your blood. You're not you're not getting your arm broken. Nothing's happening to you. But in people in face of those emotional stressors have a stress response. So we'll talk more about these as we go through the talk, and we'll especially get back to those in humans at the end of the talk. So that's kind of the definitions of what the stressors are that induce stress. And this talk is mostly about vertebrates, mostly about birds, some about humans. So then I want to talk about what happens in the condition. So what changes inside an animal when it perceives a stressor? So there are a suite of hormones that are released. The first group of hormones, epinephrine and norepinephrine. You would know these more familiarly as adrenaline and noradrenaline. Epinephrine are released from neurons throughout your body, and then also from a gland called the adrenal gland. Levels go up in your blood within milliseconds to seconds. So very rapid. And they have a suite of effects. So one of the first things that happens when epinephrine levels go up is that your liver pours glucose into your blood. And the amount of oxygen you can get in your lung increases. What actually happens is the smooth muscles surrounding your bronchioles, the tubes that go into your lungs, epinephrine causes those muscles to relax so that you can get more volume of air into your lungs. Actually, the inhalers that people use, it's a mimic of epinephrine to get their airways to open up. Speeds up the heart rate. It speeds up the force of the heart. And another interesting effect of epinephrine is that it causes blood to go to different parts of your body. So more blood is delivered to your brain and to your skeletal muscles, and less is delivered to your gut and areas that wouldn't necessarily need it. So when you think about all of these things happening, if you think about a zebra running away from a lion on the African savanna, this is going to load the blood with glucose, load the blood with oxygen, get it as quickly as possible to the tissues that matter. The next suite of hormones are the ones that I think about a lot in my life. They're the things that I am excited to work on and figure out how they work. These are the glucocorticoids. So they also have a suite of behaviors that are fairly common amongst all the different animals. Glucocorticoids also increase glucose levels in your blood. They cause your liver to push more glucose out into your blood. They also cause your muscles to break down protein, shuttle those amino acids to your liver, and turn them into glucose and pour them back out into your blood. So getting glucose into the blood is a huge piece that happens when stress occurs. Glucocorticoids also have a series of effects that are kind of at a bigger scale. So glucocorticoids suppress growth. They suppress growth hormones. They suppress reproductive behavior. In many animals, you see an increase in food intake behavior, an increase in preference for fatty and salty foods. It's been shown in lab rats. It's been shown in humans. Stress increases those cravings for that kind of food. Interesting interaction with immune function over rapid time frames. There's an increase in immune function. And then when the stressor persists, there's a suppression of how your immune system works. We can talk about that later. I talk to my students about that a lot when they wonder why they get sick when they get home from school. So when you take these all together, in general, what happens inside the body is that there's a maximizing of glucose and oxygen delivery to critical tissues, but non-critical activities are shut down. And a big point I want to make here is that these hormones that are secreted, they're found in every single vertebrate that's ever been tested, has an increase in epinephrine and an increase in glucocorticoids within seconds of perception of a stressor, and it takes a couple minutes for glucocorticoids to go up because your body has to make them once you get stressed. Every single vertebrate has these. And when we think about stress as humans, we think about ulcers, we think about type 2 diabetes, we think about heart disease. But if stress were only bad, why would this reaction be present in every single vertebrate that had ever been studied? As a side note, I'll mention this glucose is a big piece of it because when wild animals are stressed, they use that glucose to escape the situation that they're in. And that if you take away their ability to secrete glucocorticoids, those animals perish. So that's a pretty strong selection factor to keep it around. But there are also a lot of other things that are happening, especially with the glucocorticoids, that we can think about in how having this physiological response to a stressor can benefit an animal. That stress can be good and can benefit animals in challenging environments. So what I'm going to talk about is kind of thinking about how glucocorticoids may alter behavior and physiology in wild animals and how that may benefit them. And when we think about glucocorticoids, so a stressor, this is this pathway that happens when someone becomes stressed, acts on the brain. This is a bird brain. And that brain has several signals that end up sending a hormone to the adrenal gland. So the adrenal gland is usually gray, happens to be pink in this picture. It's a triangular gland that sits on the top of both of your kidneys. And that's where they are across all vertebrates. And when that adrenal gland is activated, it secretes glucocorticoids out into the circulation. And there are two main types of glucocorticoids. And there's cortisol, which is the type found in mammals, so in us as well, and in fish. And then there's corticosterone, and that's found in the amphibians and the reptiles and the birds. I tend to call them all cort, because that simplifies everything, and it's a much easier word to say. And just to remind you, corticosterone, we think of it as shutting down non-critical activities so that an animal can focus its time and its energy on self-maintenance, on things that will help it cope with the current stressor and get through that challenge. So when we think about the amount of time and energy that an animal has to spend in its days, It's not infinite, right? So animals can't live forever and reproduce and grow as much as they want. They have to take the amount of time and the amount of energy they have and allocate it across these processes. So it's a trade-off between them. And when we think about what's happening during experiencing stress, is that we think a stressor causes an increase in glucocorticoids in court, and that causes a decrease in the amount of time and energy that's spent on non-critical processes to get through that day to the next day, to shut down those processes such as reproduction and growth, and favors processes such as self-maintenance that will enable an animal to survive that challenge and move on to the next one. So what I want to do with this talk is tell you about 3 projects we've worked on in the lab. 2 that I've worked on with in the past, and 1 that we are currently working on in the lab now. So that one isn't really, I don't have data with this one. I just want to tell you about some exciting questions that we're asking that I'm excited about. And each of these are trying to help us understand, what are glucocorticoids doing? Are they causing this transfer in what animals should be spending their time and their energy on to help in survival? And how does it work? And what we're going to do is we're going to start by talking about Laysan albatross and thinking about the decision of the young to fledge. When should these young leave their parents and become independent? And then we'll spend some time talking about white-crowned sparrows that I studied for about 15 years down in the Sierra Nevada. And when they arrive at the beginning of the season, conditions are extremely variable. It's sometimes, there's huge amounts of time between they arrive and when they may be able to start breeding. And how might glucocorticoids help in that flexibility of when to decide to breed, given conditions? And then we'll finish up talking about the phenomenal mountain bluebird and what I'm looking forward to doing with some work on feathers with them. So I should get a drink of water. Okay, so Laysan albatross. I don't know how many people in here have been able to go to Hawaii to see the colonies of Laysan albatross. The work I'm going to present on these guys was done by my former graduate student, Rachel Sprague. She did her PhD in wildlife biology. She's now the assistant director for the Hawaiian monk seal recovery program in Hawaii, representing very well. She just became president of the Wildlife Society. And so that's, I'm very proud of her, and she did some great work. I'm going to present some of the stuff she did for her PhD. So these are breeding pairs of albatross. Here is one pair with an egg, and this is a young that has not yet fledged. So it is in the process of going from its downy chick to adult plumage. So the Laysan albatross nest in the Hawaiian Islands. And the large groups that you see like this are out on Midway Atoll, where they have between 200 and 400,000 breeding pair. The work that Rachel did that I'm presenting today was done on Kauai. I don't know how many people have been to the Kauai National Wildlife Refuge. It's on the northwest corner. It's got that big white lighthouse. And if any of you have watched Lilo and Stitch with your grandchildren, that White House plays a large role in those movies as well. So the population that she was studying is right next to that lighthouse. And it's amazing because it's a colony of known age, known breeding history group. So it's a really rich population to do work on. And so I want to tell you about the breeding biology, how it works when Laysan albatross decide to breed. So the pair arrive at the breeding site early in November, and during that time, the mom lays the egg. Incubation lasts through January when the young hatches, and from January to June is when the majority of chick body growth occurs here. So from June to July, the chicks are done growing in skeletal size. They don't get any bigger in skeletal size, but they do grow their wings over this last month. And during this time, wings are growing about 2 millimeters a day. So the more time they can stay in that habitat, the longer their wings can be and the more successful they are in surviving to return to breed. This last period here is extraordinarily important. The decision to fledge for these beasts is important because they're fed by their parents through all of this. Any food and water they get for all of this is because their parents fly away, get squid and fish, and barf it up into their mouths when they get back. That's the only place. If they need to start getting their own food, they have to fledge. And once an albatross leaves the natal colony, it is not seen on land again for 3 to 5 years. So it may land on the water surface to get itself food. No one has ever caught one on land until it returns to this natal colony to breed. So it's a pretty huge decision to leave. And what's happening during this is that the parents are feeding all throughout this. So if we think of condition, the amount of mass that that bird has for its body size is getting bigger and bigger. And it ends up that the albatross, the Laysan albatross, is one of those species where the nestlings get larger than the adults while they're sitting there in the nest. They can get up to 1.5 times the mass of an adult. And that what happens is as they start growing out their wings, the parents are coming less and less often, less and less predictable. So it's actually a strategy to put on a ton of mass and then use those endogenous resources to be able to fuel the wing growth as the parents come less and less toward the end of the breeding season. And that's kind of a long time, so you can imagine. The parents are ready to be doing something else by that point. So what we were interested in is how do these chicks decide to fledge? Because they're getting food. The longer they can stay there, the longer they can grow their wings. There are no natural predators for these guys. There's no reason for them to speed up and get out of town. The longer they can stay there, the longer they can grow their wings, and it's been shown longer wings mean greater chance of survival to return back in 3 to 5 years. So what Rachel did was she wanted to understand how are they balancing this lessening food availability with the need to have longer wings. So the first thing, what we have here is you're going to see a couple of these graphs. On the x-axis are the days before fledging, so day 0 is fledge. So these are the 60 days before fledge, and they're repeated measures from about 14 chicks throughout all of this. And this is wing length. So this line represents the shortest wing length we ever saw a chick fledge at. So it seems to be a morphological limitation, below which chicks don't leave. But what that suggests is that there's actually a lot of variation above that. So why is it that some chicks can stay until their wings get really long, and some have to leave when their wings are not at all as long as they could be? Well, Rachel, in these captures, also measured body condition. So this is the amount of mass divided into structural size. And this shows that decline in body condition that occurs as they get towards fledging. Their parents are visiting less and less often, providing less and less food. They're losing mass because they're using that energy to fund growing their wings and existing from day to day. So is this loss of body condition and the lack of predictability of adult visitations, is that stressful for these guys? Well, on capture, Rachel also took a blood sample, and we measured glucocorticoid levels. We measured corticosterone levels. And absolutely, there's a significant increase in corticosterone levels as these animals approach fledge. So our question was, is this elevated level have anything to do with this variation in wing growth? That maybe animals that have a greater decline in body condition have a greater increase in corticosterone, and that may mean they have to leave sooner because they have to prioritize getting food over getting their wings longer. So to look at this, we couldn't manipulate corticosterone levels in these guys. These are protected species A lot of work has been done on them. And so what we did instead was Rachel supplementally fed a subset of the chicks. So every 3 to 4 days, she fed them squid or fish that would almost cover the amount they would usually get from parents when parents were still coming fairly often. So how does supplemental feeding affect this decline in body condition, this increase in court, and does it change fledge decisions? So the first thing we have is these red data are the birds that were fed. And absolutely, supplemental feeding means they don't lose anywhere near as much weight. So we are inhibiting that loss of mass that occurs before fledge. And we're also offering a predictable every 3 days delivery of food. Does that affect corticosterone levels? And it absolutely does. You might think from looking at this that these animals would be more stressed because they're being handled to be fed. Not true at all, actually. These guys have much lower corticosterone levels, less than half. If you look at the comparison at fledge, the ones that were supplementally fed had less than half the amount of corticosterone than birds that were dealing with the decline in body mass and the fewer and fewer feedings. So then the question is, do we see any relationship between these fed and unfed birds and when they choose to leave? And we do. So these are the control birds that have the mass loss and the increase in court, and they fledge at about 150 days post-hatch. The ones we fed stayed for about 10 days longer, which when you look at the entire period, it doesn't look like that big of a change. But think about, these wings are growing 2 millimeters a day. So 10 extra days means 20 millimeters per wing, 40 millimeters more of surface area to fly on once they leave. So this is a huge increase in their capacity to succeed after they leave. Okay, so in the timing of fledge in Laysan albatross, why do we think court is beneficial? What do I think these data tell us about what court might be doing? So as chicks mature, they have to balance food and nutrient availability against flight availability. Parents are coming less often. They want their wings to get as long as possible. If food availability gets too low, the chick should fledge, become independent, and go out to get their own food. But if food is readily available, stay. There are no natural predators. Get your wings as long as possible. And we think corticosterone is mediating this balance. And if we put it back into that original trade-off that we talked about, as body condition declines and corticosterone levels go up, we think that corticosterone is shutting down processes toward growth and increasing processes that will enhance survival over the long term. So the best thing about working on albatross is since they have no natural predators, you get to walk up to them and pick them up. And they're these amazing beasts. When you put them back on their nest, the adults, their drive to incubate the eggs is so strong that you handle them, you're like holding them like this. They're about the length of a turkey, but much more narrow. And you hold their bill in their hand, because that's the part they'll get you with. So you hold them like this, and you put them down so that they're looking at their egg, and you can feel their whole body go. And when you let them go, they just go and sit on it. It's awesome. It's one of the best things I've ever done as a biologist. All righty, so that's thinking about the role of glucocorticoids in timing of fledge in Laysan albatross. The next thing I want to tell you is about a beast that's near and dear to my heart. It's the white-crowned sparrow. I started working on this bird in 1993 when I started graduate school, and I've continued working on and off. The work I'm going to tell you about today was done in the High Sierra. So this is the mountain white-crowned sparrow, the subspecies that breeds throughout the western United States in the Cascades and the Sierra and the Rockies. And so the population that I studied breeds just outside Tioga Pass. So for those of you who have been at Yosemite Park, we are the eastern border to Yosemite Park, is Tioga Pass. And this is what it looks like when males arrive in early May. So they migrate up from Mexico. Both males and females migrate up. Males arrive first. And they get there, and there's 99 to 100% snow cover. And the males start setting up territories. So conditions can change really rapidly because so we're at about 10,000 feet elevation. And when the males first get there, there's 100% snow cover. That's May 28th. June 13th, you have some snow melting. You start to get the willows coming out, and the willows are where the males build their nests. So you can see here, June 24th, the birds start to lay eggs when the snow cover's at about 50%, is when there's enough willows out, and 50% snow cover tends to vary really nicely with date of first egg lay. And then here's July 11th, when the meadow's all the way out. So conditions year to year are highly dependent on the amount of snowpack from the winter before and the amount of snowfall during the month of May. So this is a picture of one of the breeding sites on June 7th, 1997, the first year I did work here. And that's a picture of the same place, June 8th, or June 7th, 1998. So there's enormous variation in the conditions these males and females face when they get to this breeding site. And in fact, this is a graph of when in each one of these dots is a different year that I was there, and when was 50% snow cover reached, and when was the date of first egg lay. So the first thing to see is that egg lay is really closely related to snow cover. The second thing is that there is a difference of about 50 days between years when they will actually start breeding. So this environment requires a flexibility in when do you turn all those systems on and spend all your energy on producing young, and when should you not yet, because you're not going to have a place to do it or enough food to eat to get it done. So what I think about in this system is when these animals are migrating, they're highly mobile, they're eruptive, they're social, they flock. When they settle into breeding, it's a completely different life history stage. They're stationary, they're highly territorial, they're very aggressive to each other. So what's happening during this arrival phase in this bird, in this species that needs to be so flexible because conditions vary so much year to year? And in the white-crowned sparrow, this is especially important because it's a longer distance migrant. So it doesn't have any environmental cues when it departs from Mexico in the winter what conditions are going to be like when it gets there. So given that, it has to wait till it gets there to decide when does it ramp up breeding and how long should it stay in that migratory phase. So what we do, the question we're asking is how do these animals regulate this flexibility to initiate breeding? And one of the ways we study this is we put transmitters on the white-crowned sparrows. This is a transmitter. This piece at the end is about the size of the last joint of your pinky. And each one of these is an elastic loop of thread so that the transmitter itself sits on the back of the bird and one loop each goes around a leg. So there's a white-crowned sparrow wearing one. You can see the transmitter right there. And if you have a receiver and a 3-element Yagi antenna, you can find them because each bird has a transmitter that has an independent frequency on it. So I want to tell you about a couple different studies that we've done over our time working there that help us understand a little bit about what corticosterone might be doing to help with this flexibility. So the first piece I want to tell you about is so we put transmitters on birds and we just hiked around and found them a couple times a day, every 3 or 4 days for most of the season. And we wanted to evaluate when do they really settle down into the area of their territory and hang out only around their nest versus when they first get there and they're wandering all around trying to find food in the best territory. So this is the area over which they're found. So the higher numbers mean they're spending a lot more time further away. And this is 3-day cycles. Throughout the nesting period, for reference, this is when the first egg was laid. So what you can see is there's a real change in behavioral strategy as when they start and they're in arrival phase and when they get into breeding. They're found over much smaller areas right around where their nest is. So we did this same thing by following them day to day and looking to see where they were found. But halfway through, we gave half of them implants of corticosterone. So we actually experimentally increased the amount of hormone they had circulating. And what this is a map. So this is Lee Vining Creek, which runs through the middle. This is a road that comes by. This is that same creek, but a map for a different individual. And what I've shown here is how much area were they found over before we gave them an implant of hormone, before we changed anything. And so this individual was located at all these spots. This individual was over a little bit greater range. And these are just examples, but this individual didn't get any hormone at all. It was a control. And it's this green polygon, which you can see a little bit. So there's a little bit of a change, but it's fairly similar to the blue. This individual got a corticosterone implant and is found over a much greater range of space afterwards. And in fact, every individual that received a corticosterone implant increased its activity range between doubling it and getting up 900%, above 100%. So what it looks like is that as birds are in arrival phase, they're highly mobile and moving around. When they move toward breeding, they're settling into one area. This hormone corticosterone appears to be reversing that and pushing them back out into potentially more exploratory moving behavior. The second piece I want to tell you about is looking at how they respond to storms. So this is what it looks like at the field breeding site when they first arrive. In May, up in the high Sierra, snowstorms are really common. And they come in and they dump snow over all available foraging areas. These birds are eating insects that have blown up from lower elevation and died on the snow. So a little bit of snow cover decimates food availability. So when that happens, they abandon their territories that they've been defending and head to lower elevation sites, usually about 3,000 feet lower in elevation, to places that aren't anywhere near as extreme. So we followed birds up at the breeding site, and when storms came and they left, we found them down low. And we wanted to ask, was the choice to be high at the breeding site or low at a more kind of migratory staging place affected by how much corticosterone they had in their blood? So half the birds received implants of corticosterone to increase their stress hormone level, and half were controls. And we looked at how much time those birds were up at high elevation. And we have the percent of time that we were looking that birds stayed at the high elevation breeding site, and so were spending their energy and time on breeding. And control birds were there almost 75 to 80% of the time. Birds with augmented corticosterone levels were only there about 25% of the time. So in this case, it looks like when conditions become challenging, these birds that have higher cort levels are staying further in that migratory state and not coming into the breeding stage where they want to be up at the breeding site. So if we put all this together, great. So why in this case might corticosterone be beneficial for these animals? Well, environment at the breeding ground where these guys come after a long migration is highly variable, and there's no way to predict it before arriving. So these sparrows need to be flexible in the initiation of breeding, and it needs to depend on the weather present at the breeding site and their food availability. So what it looks like to us from our data is that cort appears to inhibit entry into the breeding behaviors, moving animals back into more of a migratory state until conditions up at the breeding site improve. So when we get back to this trade-off between reproduction and survival, what I've shown you is that an increase in corticosterone that may be associated with a stressor appears to decrease the amount of time and energy spent on reproduction. But I don't really have any data that I've shown you on survival. But one of my graduate students evaluated 10 years of data that we have from this population, where we have known corticosterone levels that from when we caught them in our hand and got a blood sample, and we have known reproductive success and we have known survival for those 10 years. And what we know from that analysis is that animals that are more reactive to stress, that have more corticosterone secreted in response to being captured by us, have higher rates of survival over that time. So that we do see a decline in reproduction and an increase in survival associated with this increase in glucocorticoids in this population. Okay, so the last piece that I want to talk about is just, I want to communicate to you some questions that we're starting to ask in bluebirds, and some different reasons why I'm really excited about this. A, mountain bluebirds are phenomenal. They're just one of the most beautiful species I've ever had the joy to handle. And B, they're local. So it's one of the first times I've talked to you about things I've done in Hawaii, things that I've done down in the Sierra. Linda mentioned that I've done some work in the San Juan Islands. I get to work on these guys here, and that's really nice. It makes it really easy for me to bring graduate students and undergraduates out to do the work, and it's a really nice thing that's developing. So let me explain to you the questions we're asking in these guys and how it applies to this overall question. So, and this is work that is being spearheaded by my new graduate student, Sarah Burke. She's in the second year of her PhD in the biology program. And what we're thinking about in bluebirds is the production of feathers. I've never thought about molt before, but it's a really important life history stage because it determines the success of that animal in a lot of different ways over the next year. So if we think about this trade-off, feathers are important for survival because feather structure, so the interbarb, the barb distance, the density of those barbs, how permeable that feather is to air. I know Brett talked about that a little bit last week. That's incredibly important for how efficiently those birds can fly, how well they can escape predators, how much energy they have to spend on migration. So thinking about the energy that these birds can put into the feather structure should have huge implications for their survival over the next year. On the other hand, these are sexually selected traits, and in these guys, they're a phenomenal bright UV blue. Now, the coloration in blue feathers is interesting. I can talk about it afterwards if you want. It's actually not a pigment. It's a refraction of light, so that they have to grow the outer structure of that feather with these tiny little holes in it, and if those holes are all the same diameter, then the color is brighter. If those holes are grown more quickly without as much precision, then there's a greater dispersion of the type of color that's given off, and the blue isn't as bright. So as we think about what happens when animals are challenged during molt, what happens if food availability isn't as high, weather isn't as good, and these guys don't have optimal resources when they're growing these feathers? If there's a stressor that occurs, I would expect that glucocorticoids would favor enhancing feather structure over feather color, but we don't know yet. So that's what we're working on now. Sarah's asking a lot of questions actually about the sexual selection of these bright colored traits and interaction between males and females. This is what I care about, is being able to figure this out. Okay, so with these number of examples, I hope I've gotten you to think a little bit at least about how stress, which we all think of as this horrible thing, can actually benefit animals in the wild, can benefit the timing of large life history transitions into dependence, can help with flexibility in initiating breeding. But at the beginning, I tended to stress out some people over here, and I did promise I would get back to stress in humans just a little bit. So, yes. What about stress in humans? And the issue with humans is that, with people, is that we have emotional stressors that we turn on over and over and over again. And as you know already, epinephrine glucocorticoids are turned on every time you turn on that emotional stress response. And if you have to have a bunch of glucose and oxygen in your blood to run away from a lion, that's okay. If you are pouring glucose into your blood repeatedly and then doing nothing with it, that leads to all sorts of things that aren't as good for health. So what I want to talk about is just a little bit of thinking about approaching with a little bit different psychology as things become stressful. and thinking about the role of personality and temperament in dealing with stress. And I'm going to recommend you to a book written by a professor at Stanford called Why Zebras Don't Get Ulcers. This is written for the general public, and he's a really funny writer. It's very interesting to read. And what it's about is all those things that I told you about that happen during a stress response that we think about happening in animals. How do those get turned into long-term stressors and disease in humans? And then what I'm going to tell you about now is what happens in the last two chapters here, which is how can you cope with it? What kind of things can you think about? So just to finish up, I just want to mention a couple things which affect the way people respond to stress. So one of the big things is how much control do you perceive that you have? So imagine walking into a room full of people you don't know. Some people walk into that room and think about all the things that could go wrong. And some people walk into that room and see opportunity. And you can see yourself being one or the other of those people. The more control you perceive that you have, the less glucocorticoids you secrete. It's been shown in a bunch of different studies. So one thing that Sapolsky recommends is that you start working on looking for ways to find control in stressful situations. You walk into a room, what can you do to help you feel more in control of the situation? And that the more you work on it, it actually helps feeling more in control and secreting less glucocorticoids as things seem more out of control. It's actually one of the main reasons why people prefer to drive than fly. So driving death rates are much higher than they are getting on a plane. But people behind the wheel perceive themselves to be in control. And so they would much prefer and are much less worried about driving than they are about flying. The second piece here is predictability. If bad things are happening but they're predictable, much less cort is secreted in response by people. So one of the ways I incorporate this into my life is actually with my students. So in the class that I teach, I give them practice tests. I let them know what's coming. I go everything by the book, and I actually give them more control. We have multiple exams, and they get to drop one so that they can choose, is this the one that I'm going to not do as well on? It puts them more into a place of control where they are less stressed out by what's happening in that classroom. So things that if they can be predictable, even if they're a challenge, will cause less glucocorticoids to be secreted. Having an outlet for frustration helps a lot. So having, in rats, they've done studies where if a rat is being stressed, if it can chew on a peg, it has less cortisol secreted. If it has a smaller rat to bite, it has less cortisol to be secreted. Sapolsky calls those people who give ulcers instead of getting them. So thinking about having exercise, having some way to have an outlet for your frustration, reduces cortisol levels. The last one here is social support, and this is a huge one that actually helps people reduce cortisol levels a lot by having people to talk to, having friends to have coffee with, having people to be able to share things with. So when you have a phenomenal amount of chronic stress going on, some recommendations that Sapolsky makes, be physically active. At this point in 2015, this might sound trite. Everyone's telling you to exercise. Everybody, get out for 15 minutes a day. Ends up, a couple different things are good about it. One is that when you're stressed out, you're pouring glucose into your blood, and you're not doing anything with it. And if your body can use that glucose and reset the level down at a lower level just by a 15 to 20 minute walk, you are reducing your risk of type 2 diabetes by a ton, because it's that continuous mobilization of glucose sitting there in your blood. Your body then has to store it back up with insulin. Your cells become less sensitive to insulin. You have to secrete more the next time. You're still getting glucose into your blood. Go for a walk. For 15 or 20 minutes helps reset your glucose levels every day, even just 5 days a week. Another reason that it's good is that working through problems in your head with movement actually has been shown to put problems through different pathways in your brain and can help you solve them in different ways. Having social support, again, is a huge piece in dealing with chronic stress. Taking care of your body, I tell my students, try to get enough sleep, try not to drink too much alcohol. that is a good thing to do to help yourself deal with the stress that's happening. Relaxing is a great thing to be able to do if you can find the time. One thing to do if you can't find the time. So this is a fun trick. Where you're sitting, and I don't imagine any of you are very stressed out right now, but breathe in really deeply as you can, and hold it for about 5 seconds. So that expansion of your rib cage activates your parasympathetic nervous system. Adrenaline, epinephrine, is secreted from your sympathetic. That's the active one that's way turned on in me right now. If you want to shut that off and allow your body to relax, you can activate your parasympathetic. And you can actually trick your body into doing it by expanding your rib cage out. I had my mom do it on my wedding day. She needed to relax. So it's a fun thing to do if you want to get yourself, to help just get yourself feel calmer. And lastly, having an emotional outlet. Having a counselor to talk to or having social support are great ways to help with chronic stress. Okay, I'd like to thank the 2 graduate students whose work I talked about a little bit today. I had funding from the National Science Foundation and a lot of support from the University of Montana, and I'd be happy to take any questions. And thank you for putting up with all of that. I feel better already. Oh, good. Yes. Yeah.
I have a good question about time. I'm a graduate student and I am experiencing a lot of stress, and I was wondering if there are any correlations you found between the amount of times or episodes of stress more frequently.
I can repeat what you said.
More frequently, if that had any impact on growth, production, reproduction, anything like that. So.
Right. So the question was, how much does maybe how often stressors happen, or how much time over which stress is happening, affect reproduction and growth and all these things? I'll answer it first from a human perspective. As a graduate student asking, I understand.
Or as a bird.
Or as a bird, right. Well, so one of the interesting things, so when we think about, I was talking about acute stress, which are stress happening over short periods that animals cope with and then move on. Chronic stress is stress that lasts much longer and actually causes significant changes in physiology because it lasts so long. And that's when you get into more health problems. The problem is that repeated acute stressors have the same effect as elongated chronic stress. So people who turn on their stress response repeatedly face the same health problems as people who are facing a longer chronic issue. In terms of wild animals, chronic stress is not as much of an issue in wild animals because most chronically stressed animals become part of the food chain really quickly. The only situation I know of where chronic stress persists is in social hierarchies, dominant-subordinate interactions. The subordinate animals are often have chronic stress occurring. Sapolsky has some great work. He studied baboons for decades and looked at that. And as far as the timing, I think that when the stressors last longer, the response becomes more extreme until it starts to shut off because it's too much. So chronic stress, then that causes all sorts of problems as well. Yeah?
Isn't a repeated drawing of blood from a bluebird pretty stressful? And how do you get blood out of a bluebird?
Absolutely. So we take blood from the same vein you get it taken from, which is the alar vein. It's the vein that runs through your arm coming back up here. And it's very nice working on passerines, on the small perching birds, because you can hold them in your hand. So what we found is that we can get, and we're actually taking very small levels of blood. A cool side note is that back in the 1970s, if anyone wanted to study the hormone levels in a wild animal, they had to shoot it because they had to get enough blood to be able to identify the hormone levels in that. And so you couldn't just take a sample and let them go. And in the 80s, a guy named John Wingfield at the University of Washington figured out how to make the assay more and more and more sensitive so that you need less and less and less blood to get the amounts. So I can measure corticosterone levels from 5 microliters of plasma, which is hard to picture even. So I can get, and I like to get a sample within 3 minutes of capture, which would show baseline non-stress levels, because it takes a couple minutes for that bird to produce them. And then I get levels at 10 minutes and at 30 minutes to look at their signature of how they respond to stress. The smallest animal we've ever taken blood from are developing zebra finches. So day 4 nestling zebra finches, and those weigh, I think, about 5 grams. And you just get a drop or two. And they actually recover from it. People have done studies that holding a bird and taking a blood sample, they tend to get back to near baseline levels within about a half an hour. Yeah. Yeah.
In humans,
Yes.
sleep deprivation, how does that impact cortisol if you have a bad night?
Yeah, well, I'll see if I know anything based on fact. Right, exactly. Well, so sleep deprivation throws everything off. It throws homeostasis off. So your ability to regulate different processes goes way out of whack. And do I know anything about cortisol? I expect cortisol secretion goes up with sleep deprivation. I don't know of any data on it, on how it affects stress responses or if levels go up higher. So I can't help you with that answer. I could try, but I should stop. Yeah.
On the albatross, when they fledge.
For the albatross, when they fledge, do they just all of a sudden one day take off and go away? They don't do any, well, maybe I'll try it and...
They, it's pretty awesome. They are walking around more and more and mixing with other of the juveniles, and they'll do some practice taking off, and they'll float a little bit and stop, and they'll float a little bit, and then one day they just disappear. It depends on, so the population we're studying nests on a cliff, so they can actually take off and go somewhere. The Midway Islands, the Midway Atoll, it's a flat area that goes directly out into the sea, and there are predators that wait for fledge time out in the first 50 meters of ocean, because many of the chicks don't make it and fall in. If they fall close, they can walk back and try again. But it doesn't tend to be a process where they come back and forth a lot. It's a fairly phenomenal switch. Yeah. We have one? Yes?
Well, I was just wondering, when you do add the court with the white crown, do you interrupt the time that they might do reproduction? And is that
How does
a
it
negative
affect
thing?
that? So we tend to, the work that I've done is always during the arrival phase. And we tend to be finishing as egg laying is happening. And then we remove the implants and let the birds go on about their business. So, and the birds that we've implanted return the next year as often as ones that haven't been. If anything, I think we may delay reproduction a little bit on them, but those birds have successfully nested afterwards. So yeah, for a long time the population was being followed. The population actually was started to be studied in 1967 by a guy named Marty Morton out of Occidental College, and he had data on them every year. I joined the group in 1997, and it was about 5 years before I did any hormone manipulations at all, because we were using it as a long-term database about survival. And after that, I just kept track of the ones that I had done implants on. So, yeah.
There is a study in the Journal of Sleep, 1997.
Gosh, some data!
Sleep loss results in an elevation of cortisol levels the next evening.
Oh, so a bad night of sleep elevates cortisol the next evening, and then I wonder how much time it takes to recover. One thing about your natural cortisol levels is that there's this baseline. Cortisol is always secreted at low levels, and it actually helps with activity and nutrient regulation in the body. So levels are low during the day, and then they rise while you're sleeping. And they peak right before you wake up. if you're on a regular schedule. In animals that are on light-dark cycles where it's known, it's very easy to follow. And they think actually that peak is supposed to help with that surge of activity that happens when sleep ends. And it happens in mice and rats that are nocturnal. It goes up during the day while they're sleeping and peaks just before the active period at night. So I would imagine it's higher then because that's when it's starting to go up, but it's even elevated above that. Thank you.
Thanks for not turning off your phone.
Yeah, right, exactly. All righty. I think we're good. Thank you all for coming.